Okay, let's unpack this. Today, we're diving deep into, well, one of the most fascinating and honestly rapidly evolving frontiers in health. We're talking about the intricate connection between your gut and your brain.
Flame.
Imagine like a super highway of communication, constantly linking your digestive system directly to your central nervous system.
Yes.
And it's influencing pretty much everything, right? From your mood to how you think.
Absolutely. And what's truly groundbreaking here is how this bidirectional system, the gut-brain axis, as it's known, actually operates. It's not just simple digestion, It's this really complex interplay. You've got neuronal signals, immune pathways, metabolic stuff, all working together. And it profoundly influences mental well-being and even the progression of neurodegenerative disorders. This understanding, it just opens up entirely new avenues for potential therapies. It's quite exciting.
Yeah, it really is. And you've shared some incredible sources with us for today's deep dive. We've got everything from pioneering research on fecal transplants, which we'll definitely get into, to the surprising power of personalized diets, and even these tiny molecules that act like messengers between the gut and brain. So our mission today is really to extract the most important nuggets of knowledge from all this, explore some surprising facts maybe, and help you understand what this all means for your health.
Exactly. And this deep dive will explore, well, both the established findings and some really cutting-edge innovations. We'll be looking at how understanding these connections might offer genuinely novel therapeutic approaches for conditions that, let's face it, have historically been incredibly difficult to treat.
Okay, so let's start with the basics then. This gut-brain axis, we're saying it's a communication system. But when we say communication, what exactly is happening? How are these two seemingly separate parts talking to each other?
Well, it's far more than just that feeling of butterflies in your stomach, although that's part of it. Classically, the system relays information about, say, nutrition, gut movement, motility, and feelings of fullness, distension. But recent studies, wow, they show its influence is much, much broader. It's affecting things like how brain support cells, microglia, develop and proliferate, how immune cells, T cells traffic around the body, inflammation levels, obviously, even processes linked to insulin resistance and the buildup of proteins associated with diseases like Alzheimer's amyloid deposition, synapse formation too, how brain connections are made.
That's huge.
It is. I mean, think about experiments with germ-free mice. These are mice raised without any gut bacteria from birth. Their brain structure and function are actually abnormal. And it's strongly correlated with that lack of gut microbiota, particularly in the postnatal period. It just underscores how dependent the brain is on these signals coming from the gut right from the start.
And who exactly is sending these signals from the gut end? You mentioned microbiota.
We're talking trillions of microorganisms, your gut microbiome. It's typically home to over 1,000 different species of bacteria, usually dominated by two main groups, Bacteroidetes and Firmicutes. But here's where it gets really interesting, and you touched on this, the dynamism.
Right, you said it's incredibly dynamic. So what makes it so fascinating? How much influence do we actually have over who's living down there?
It's fascinating because it's not static at all. It's constantly changing based on your diet, your lifestyle, medications you might take, your overall health, even stress. And what's really striking is how quickly it can shift. We're talking about a living, evolving ecosystem inside you. And the key thing is you can actively influence it.
Okay, so influencing it sounds good. But what happens when things go wrong? When that delicate balance gets disrupted? Researchers call it dysbiosis, right? It turns out this imbalance, it's being linked to a really surprising range of conditions way beyond just tummy troubles.
Yes, dysbiosis. And this is one of the most, I'd say, profound insights from the sources we looked at. Gut imbalance isn't just about digestion. It's now considered a key suspect in major neurodegenerative diseases. There are direct associations shown between dysbiosis and conditions like Parkinson's disease, Alzheimer's, multiple sclerosis, even ALS.
Wow.
Yeah, it completely reframes how we might need to approach these really devastating conditions, looking beyond just the brain itself.
And it's not just the major neurological diseases. You mentioned mental well-being too. Dysbiosis linked to depression, anxiety, schizophrenia, even autism. Is there like a common thread here, a mechanism that explains how messing up your gut bacteria can affect such different things, from Parkinson's to depression?
That's the million-dollar question, isn't it? And yes, there are converging ideas. If we try to connect the dots, the gut microbiota seems to influence mood and brain function through several key pathways. Let's break them down. First, there's neurotransmitter synthesis.
Okay, brain chemicals.
Exactly. Your gut microbes actually help make vital neuroactive compounds. Get this. Roughly 90%, 90 of your body's serotonin, the feel-good hormone.
90%.
Yeah. And about 50% of your dopamine, they're produced in the gut. And the byproducts of microbial metabolism, these metabolites, they influence how much is available and how the signals work. So when dysbiosis happens, it can directly mess with this production line.
So an imbalance down there literally changes the chemical messengers your brain relies on for mood. That's incredible.
It is. Okay, second pathway. The stress response, specifically the hypothalamic pituitary adrenal axis, the HPA axis.
Right, the body's main stress system.
Precisely. Gut bacteria significantly influence this axis. Dysbiosis can actually lead to HPA hyperactivation, basically. It ramps up the stress response. This means increased cortisol levels, which is a really common feature in major depressive disorder. In fact, studies where they deplete gut microbiota show this hyperactivation of the stress axis, particularly at certain times of day. Shows how tightly linked they are.
Okay, neurotransmitters, stress response. What else?
The third big one is inflammation. An imbalance in gut bacteria can trigger chronic low-grade inflammation. These bad bacteria, or just an imbalance, can cause the release of pro-inflammatory cytokines. These are signaling molecules that basically shout inflammation throughout your body, and that includes reaching the brain. This systemic inflammation can disrupt neurotransmitter function, it can impair neuroplasticity, the brain's ability to adapt of the gut, and that contributes directly to symptoms of depression.
That's a really compelling picture. Neurotransmitters, stress, inflammation, all influenced by the gut. And it seems like real-world studies are backing this up, too. Our sources mentioned a fascinating study on new prison inmates. It showed a clear link. Compared to controls, the inmates had significantly higher anxiety and depression scores.
That's right. And crucially, their gut microbiota looked different. It was more dominated by a group called Bacteroids, whereas the controls had more Prevotella. The inmates also had much lower levels of bacteria genera, known for producing those beneficial short-chain fatty acids we'll talk about later. And interestingly, they had enriched genes and pathways involved in making neurotransmitters, like the shikimate pathway. It really suggests the gut microbiota shifted alongside their mental state.
Okay, so the gut environment changes, and maybe that influences the brain. But what about things getting from the gut to the brain more directly? This idea of leaky gut, leaky brain comes up. What's that about?
Right. This raises important questions about direct pathways and barrier integrity. Both your intestinal lining, the gut barrier, and the blood-brain barrier, they're designed to be selectively permeable. They're gatekeepers, controlling what gets in and out. But if the gut barrier is compromised, becomes leaky, then things like toxins or even bits of microorganisms can breach that lining. They can then get into your systemic circulation, your bloodstream.
And potentially reach the brain from there.
Potentially, yes. And microbial interactions with the blood-brain barrier itself are being studied. There are different ways this might happen. Things could pass through the barrier cells in a little bubble, a vacuole that's transcytosis. Or they could slip between the cells through the junctions parasitosis. Or, and this is quite clever, they might even hitch a ride inside one of your own host cells, like a Trojan horse.
Wow. So these barriers aren't totally foolproof.
Not entirely. And this concept of leakiness from microbial metabolites, those chemical byproducts and other compounds, is really emerging. It suggests that potentially harmful substances can gain access to the brain, contributing to inflammation there and disrupting neurological function. Though it's fair to say the exact mechanisms and the overall leakiness concept still need more detailed explanation and maybe a more unified framework.
Okay, so let's connect this back to specific diseases you mentioned earlier, like Parkinson's. The sources suggest something about misfolded proteins starting in the gut.
Yes, for Parkinson's, there's a growing hypothesis that the hallmark misfolded protein, alpha-synuclein, might actually originate in the gut and potentially travel up to the brain via the vagus nerve, that major nerve connecting the gut and brain.
Like climbing a ladder straight to the brain.
Sort of, yeah. And here's a really surprising finding from the sources. Some types of gut bacteria actually produce a protein called curly. And structurally, curly is quite similar to that misfolded alpha-synuclein. So the idea is maybe this bacterial protein could act as a trigger, initiating the misfolding process that characterizes Parkinson's.
That is wild. What about Alzheimer's?
For Alzheimer's, the focus is a bit different, but still involves the microbiome. Research points towards altered bile acid profiles in Alzheimer's patients, specifically an increase in certain types of secondary bile acids, which are produced by gut bacteria and can be cytotoxic, harmful to cells. This strongly suggests that the gut microbiome plays a direct role in the progression of Alzheimer's disease too, possibly through these toxic metabolites.
Okay, this is all pretty sobering, seeing the potential negative impacts. But the flip side is opportunity, right? If the gut has this much influence, maybe we can influence it for the better. So what can we actually do to nurture this powerful connection? Our sources revealed some exciting strategies starting really small with tiny molecules produced right there in the gut.
Absolutely. Let's talk about short-chain fatty acids, or SCFAs. These are produced when your beneficial gut bacteria ferment dietary fibers, the stuff you eat that you can't digest yourself. And these aren't just waste products, they are active signaling molecules, chemical commands, really. They influence everything from your brain's energy supply to your mood.
Okay, so what are the main ones?
The three major players are acetate, propionate, and butyrate, and each one has unique beneficial effects. Acetate, for instance, is an important energy source directly for brain cells. It also has anti-inflammatory properties, can help reduce appetite, and protects the gut barrier by tightening up the junctions between cells.
Okay, energy, anti-inflammatory, gut barrier.
Yeah.
What about the others?
Propionate helps regulate the release of gut hormones like PYY and GLP-1. These are crucial for signaling satiety, feeling full, and regulating appetite. Getting propionate delivered to the colon has even been shown to reduce weight gain in studies. It also reduces inflammation and helps maintain the gut barrier, partly by increasing mucus production.
And the third one, butyrate. That one seems to get a lot of attention.
It does, and for good reason. Butyrate is a particularly potent anti-inflammatory. It's crucial for regulating gut permeability, keeping that barrier strong. And it can even modify gene expression through epigenetic mechanisms, basically switching genes on or off without changing the DNA sequence itself. What's particularly exciting for the gut-brain axis is the hypothesis that butyrate might increase serotonin production in the colon. And research in rodents suggests it has antidepressant-like effects, possibly by boosting brain-derived neurotrophic factor, BDNF.
BDNF, right? That's like Miracle-Gro for brain cells.
Exactly. It promotes the growth, survival, and adaptability of neurons. So boosting it is generally seen as a very good thing for brain health and mood. And these little SCFAs, they even talk directly to the vagus nerve, sending those signals straight up to the brain.
It's remarkable, these tiny molecules having such wide-ranging effects. But beyond nurturing the bacteria that make these molecules, what about adding beneficial microbes directly? Probiotics and prebiotics come up a lot.
Indeed. These are really at the forefront of microbial interventions. So probiotics are live microorganisms, like beneficial bacteria you find in yogurt or supplements. And prebiotics are non-digestible fibers. Think chicory root, onions, garlic, that specifically feed these beneficial bacteria, encouraging them to grow.
And is there solid evidence they actually help with brain-related issues, like mood?
There is growing evidence, yes. One source highlighted a comprehensive meta-analysis which pooled data from many randomized controlled trials. It found that probiotics demonstrated a significant reduction in depression symptoms and also a moderate reduction in anxiety symptoms in people who were clinically diagnosed.
Interesting. Did it matter which probiotic was used?
Well, interestingly, both single-strain and multi-strain probiotic formulations showed benefits, though there was a hint that single-strain probiotics might have shown the strongest effect in that analysis. And for depression, the dose seemed important. A dose of at least 1 billion, that's 1 by 109 colony-forming units, or CFUs.
That's the measure of live bacteria, right?
Exactly. That level or higher was associated with significant symptom reductions.
Okay, so probiotics look promising for depression and maybe anxiety. What about prebiotics, the food for the good bugs?
In that same meta-analysis, prebiotics didn't actually show a statistically significant effect on depression. Although there was a trend towards reducing symptoms, it just didn't reach statistical significance. For anxiety symptoms, some beneficial effects were noted, but the researchers concluded more research is really needed to substantiate those findings for prebiotics specifically.
So if probiotics help, and prebiotics feed the good bacteria, what does this all mean for what you should actually put on your plate? It really seems to hammer home that idea. What you eat deeply impacts your gut, which then impacts your brain.
Precisely. The evidence we reviewed consistently points in one direction. Increase the intake and, crucially, the variety of plant foods in your diet. Lots of fiber there for the microbes. And at the same time, reduce or ideally eliminate ultra-processed foods. Really, the dietary recommendations for mental and gut health align perfectly with those for overall physical health. Think about something like the Mediterranean diet, high in whole grains, fibers, fruits, vegetables, fermented foods, fish.
Right, that diet comes up again and again for health benefits.
It does, and it's been specifically linked to improved mental well-being in studies. Plus, from a practical standpoint, it's generally a cost-effective strategy. In the long run, manipulating your microbiota through sustainable dietary changes might actually be more feasible and certainly economically cheaper than relying solely on probiotic supplements.
That makes sense. Build the ecosystem through food first.
Mm-hmm.
Okay, we've talked diet, supplements, but what if the imbalance is really severe?
Yeah.
Or we want a more radical reset? This brings us to one of the most innovative and, let's be honest, kind of surprising interventions mentioned in the sources. Fecal microbiota transplantation, or FMT.
Yes, FMT. It certainly captures attention, doesn't it? But the science behind it is straightforward, conceptually at least. It involves taking a fecal solution processed stool from a carefully screened healthy donor and introducing it into the recipient's intestinal tract. The whole aim is to basically reset the recipient's gut microbiome, restore a healthier balance, and by doing that, positively influence these neuroinflammatory and neurodegenerative pathways we've been discussing.
And is there evidence it actually works for these brain conditions?
The evidence is still emerging, but it's promising, particularly from preclinical studies and some early clinical trials. Studies indicate potential to alleviate both motor and cognitive deficits in Parkinson's and Alzheimer's diseases. For Alzheimer's specifically, some studies have shown FMT can reduce those amyloid beta plaques.
A hallmark protein buildup.
Exactly, and improve cognitive performance in animal models. And some early human studies are suggestive too. Interestingly, transplants from younger donors often seem to offer additional advantages, likely due to their generally more diverse microbiota. There's also evidence suggesting it can lower neuroinflammatory markers in multiple sclerosis models, and even enhance respiratory and neuromuscular functions in models of amyotrophic lateral sclerosis, ALS.
That sounds incredibly promising, especially for conditions that had so few effective treatments. But the practicalities, how is this actually administered? And safety, that must be a huge concern.
Absolutely. On administration, the fecal matter can be prepared in different ways, fresh, frozen, or even lyophilized, which is freeze-dried into a powder, often put in capsules. Efficacy can vary slightly by preparation. For treating severe Clostridium difficile infections, which is where FMT is most established, fresh FMT shows about 93% efficacy, frozen around 88%, and lyophilized about 83%, still very high. Delivery routes also vary. It can be done via tubes inserted through the nose down to the stomach or small intestine, nasogastric or nasojejunal, or via endoscopy either from the top end, like an EGD, or the bottom end via colonoscopy, or sometimes as a retention enema.
Okay, multiple ways to do it, but safety and screening must be paramount.
Paramount. Donor selection is incredibly rigorous. It involves extensive screening of medical history, social history, lifestyle factors, plus comprehensive blood and stool tests to rule out any potential pathogens, bacteria, viruses, parasites, fungi. The list is long.
Right. So, assuming it's administered correctly with a screened donor, what are the downsides? The risks? I mean, while the potential is huge, what are the scientific community's biggest concerns or debates around FMT right now?
That's crucial. While it's often well tolerated, there are potential adverse effects. Short term, most are mild to moderate gastrointestinal issues: bloating, abdominal pain, diarrhea, constipation sometimes, pretty common after the procedure. However, more severe complications, though rare, have occurred. These include things like fever, hematochezia that's passing blood in the stool, sometimes worsening of pre-existing inflammatory bowel disease. There have been isolated reports of serious infections like gram-negative bacteremia, even bowel perforation, and tragically, even death linked to FMT, often involving multi-drug-resistant organisms from inadequately screened donors in the past.
That really underscores the need for rigorous screening.
Absolutely. And then there are the potential long-term concerns, which are still being actively researched. Could transferring someone else's microbiome increase the recipient's long-term risk of things like obesity? Or developing immune-mediated disorders? Conditions like immune thrombocytopenia, rheumatoid arthritis, IBD, IBS have been potentially linked, though causality is hard to prove. So yes, the promise is definitely there, especially for tough conditions. But caution, rigorous protocols, and much more long-term study are absolutely essential.
It's a fascinating area, balancing that potential against the risks. Okay, so we've covered diet, SCFAs, probiotics, prebiotics, even FMT.
Yeah.
This field is clearly buzzing with innovation. Looking ahead, what else is on the horizon? What are the next generation approaches for influencing this gut-brain connection?
Well, the future looks towards even more precision and sophistication. Things like synthetic microbial communities. Instead of a whole fecal transplant, designing specific cocktails of beneficial microbes tailored for a particular condition. Or engineered probiotics, genetically modifying beneficial bacteria to produce specific therapeutic compounds like anti-inflammatory molecules or neurotrophic factors right there in the gut. And artificial intelligence is playing a huge role too.
AI and the microbiome, how does that work?
AI can analyze the incredibly complex data from microbiome sequencing, identifying all the bacteria, their genes, their potential functions. One recent pilot study you mentioned showed that an AI-driven personalized nutrition program helped enhance gut microbiome diversity and reduce diet-related health risks. Participants notably cut down on carbs, protein, overall energy, alcohol, sweets, fast food based on AI recommendations tailored to their specific microbiome and health data. There are also specialized AI tools being developed like gut-brain modules, GBMs, and gut metabolic modules.
GBMs and GMMs, what do they do?
These are advanced computational models. They analyze microbiome data to predict the neuroactive metabolic potential of someone's gut microbiota. Basically, predicting what kinds of brain-influencing chemicals a person's specific gut ecosystem is likely producing. This could truly revolutionize the field of psychobiotics, allowing for highly precise, personalized interventions based on an individual's unique gut profile.
Tailoring interventions based on AI analysis. That really does sound like the future. So we've covered a lot of ground, a lot of really promising leads, but it also sounds like there's still some pretty significant hurdles to clear before many of these become mainstream solutions, right?
Absolutely. Despite all the exciting findings, significant challenges definitely persist. We already touched on the complexities of rigorous donor selection for FMT and the need for more long-term safety data. There's also inconsistency in clinical outcomes across different studies. People's microbiomes respond differently. There's huge variability. Another major challenge is that we still don't have a universally agreed-upon definition of what constitutes a healthy gut microbiome. Does it look the same in an infant, an adult, an elderly person? Probably not. How does it vary across populations? And we also need to better understand sex differences. We know microbiota composition can differ between males and females, and host responses to interventions might differ too. That needs much more study.
So given those challenges, what's most crucial for moving this whole field forward? What do researchers need to focus on?
Well-designed, large-scale clinical trials. That's absolutely essential. We need robust validation for interventions like FMT, especially for these complex neurodegenerative diseases. This means establishing standardized protocols for donor screening, for preparation, for administration, so we can compare results across studies reliably. We need to refine those donor selection processes even further. And critically, we need to deepen our understanding of the precise molecular mechanisms. How exactly are these interventions working at a biological level? It's also going to be crucial to move beyond just cataloging who is there, which bacteria are present. We need to integrate multi-omics approaches.
Multi-omics. You mean
studying the whole picture, not just the genes, genomics, but also the RNA transcripts, transcriptomics, the proteins, proteomics, the metabolites, metabolomics, all of it together. This gives us a much better understanding of the overall functionality of the microbial community, not just its composition. What is it actually doing?
That makes sense, looking at the whole system in action. So wrapping this up, what we've really unpacked today feels revolutionary, doesn't it? This whole way of looking at our health. The idea that these tiny organisms living inside our gut could have such a profound, direct impact on our brain, our mood, and even our risk for devastating diseases like Parkinson's and Alzheimer's. It's kind of mind-boggling, frankly.
It really is. And if you connect it to the bigger picture, it just underscores the incredible interconnectedness of our body and mind. It offers completely new avenues for understanding, potentially preventing, and maybe one day effectively treating conditions that affect millions and millions of people globally. It truly represents a paradigm shift in how we approach health and wellness.
So maybe the next time you make a choice about what to eat, or maybe how you manage your stress, it's worth considering those trillions of microscopic residents in your gut. They're listening, they're reacting, and as we've heard today, they're intricately connected to your deepest thoughts and feelings. Really embracing this gut-brain connection, understanding it, nurturing it, could that be the ultimate shortcut to a more well-informed and maybe even a more fulfilling life?